Bismillahirrahmanirrahim. Assalamu alaikum. I'm Husna Mohsin, MPhil scholar from Department of Zoology, Cohort University of Science and Technology. Here I'm presenting my research topic, which is molecular detection of mastitis and its impact on hematobiochemical parameters and milk leukocyte count in buffaloes of district Diyai Khan under the supervision of Dr. Kalimullah, assistant professor in our zoology department, and Dr. Kamal Shah from Faculty of Veterinary and Animal Sciences, Gomal University, Diyai Khan. Presentation outline. This is how I'm presenting my research work. First, I will give brief introduction of my research work that will be followed by the gap analysis. Then we'll talk about the main objective, molecular methods that will used in my research work. And later we will talk about the result and discussion, conclusion and recommendation, acknowledgement and references. Introduction. In Pakistan, buffaloes are the common livestock, playing an important role in national economy by producing milk, meat, and primary source of motive power for agriculture and rural transportation. Buffalo contribute around 68% milk, followed by the cattle, which is 27%, sheep, goat, and camel, which is 5% of total milk produced in the country. Pakistan is the world's second largest producer of buffalo milk after the United States. Mastitis is an inflammatory condition of mammary gland of lactating animal that lead to severe decrease in milk production, resulting in massive economic losses to the dairy industry. Bovine mastitis can be caused by the physical or chemical agents, but most of the cases is infectious and usually caused by bacteria. In mastitis, there is a break in blood-milk barrier along with impaired synthesis and secretory activity of the other epithelial cell, which alter the level of the most component of the milk. The change in hematobiochemical parameters and milk leukocyte count used is an important indicator of the physiological or pathological state of the animals. Hematobiochemical changes in the blood profile may lead to variation in RBCs, TLC, WBCs, hemoglobin, PCVs, and for biochemical parameters such as serum glucose, serum total protein, serum albumin, and creatinine alteration in mastitis infected animals. Study gap. In Pakistan, only one study has been done in Okhara and Sahiwal, district of the Punjab, whereas no research work has been conducted on hematobiochemical profile and milk leukocyte count in clinical and subclinical form of mastitis in buffaloes in Khyber Pakhtunkhwa. Therefore, the present study was designed to determine the hematobiochemical profile and changes in the milk leukocyte count in the buffaloes affected with mastitis. Objective. First objective of our research work was to find out the prevalence of mastitis and milk leukocyte count in buffaloes of study area. And second objective was to determine the impact of mastitis on hematobiochemical profile of the mastitis affected buffaloes. Methodology flow chart. Ethical approval. Ethical approval were obtained from the ethical committee of the CAST, study area and study population. The present study conducted in district Diyai Khan and study population were buffaloes. Sample collection. The sample collected from the jugular vein of buffaloes in anticoagulant blood container and transported to Faculty of Veterinary and Animal Sciences of Gomal University, Diyai Khan. California mastitis tests were also used to count the differential leukocytes in the milk, which were calculated by the standard counting method. Somatic cell count. The milk sample were also submitted for the somatic cell count and isolation and molecular detection for the identification of the bacteria. Hematobiochemical analysis and statistical analysis. Results. All prevalence of S. aureus bacteria, which is responsible for the mastitis. Total 98 samples were collected from the buffaloes of Diyai Khan, in which 26 were positive, and their prevalence rate is 0.05. The main values of hematological parameter of control and mastitic buffaloes. For hematological analysis, the collected sample of mastitis affected buffaloes who are tested for the hemoglobin, PCVs, transient erythroblastopenia count, total leukocyte count, granulocyte, lymphocyte, and the monocytes, in which granulocytes amounts are very high in clinical mastitic buffaloes, and the monocyte amount are very low. Values of biochemical parameters of control and mastitic buffaloes. Different parameters were also checked from the serum sample, and it is used, and these parameters are checked to the biochemical parameters through the automated blood chemistry analyzer, in which the alanine transaminase, aspartate aminotransferase, total protein, calcium, phosphorus, chlorine, and sodium were checked, in which chlorine amount are very high in the clinical mastitic buffaloes, and total protein amounts are very low. Main values of milk electric conductivity, somatic cell count, and control and different leukocyte count of control and mastitic buffaloes. To check the leukocyte, different leukocyte, these tests are performed in which different leukocyte were checked. And clinical mastitic buffaloes, the neutrophils amount are very high, and the monocyte amount and the macrophages are lower than the neutrophils count. Conclusion and recommendations. Regarding the prevalence of Staphylococcus aureus, the disease mastitis is highly prevalent in the buffaloes of the ICAZ. The milk leukocyte count is considered the best biomarker for the inflammatory reaction and other infection. The increased number of milk leukocyte is present in a present study could be due to microbial infection in other, as these are the major part of the immune response of the animal. It was concluded that the California mastitis test, electric conductivity, and somatic cell count were ideal for early detection of subclinically infected quarters and aid in the selection of dairy animals for either segregation or therapy. Acknowledgement. I will be very thankful to my supervisor, Dr. Kalimullah, Assistant Professor in Department of Zoology ICAZ, and Dr. Kamal Shah, Assistant Professor of Veterinary and Animal Sciences, Gomal University, DI Khan. And thanks to all faculty members and lab fellows. References. Thank you.